Okay, so I think I was sort of charged with the idea of talking about different therapeutic approaches for KCNT1. And just as a heads up, I have been involved with clinical trials for a few different disorders over time. So hopefully by the end of this little brief discussion, everyone will be able to understand what the current treatment landscape looks like for KCNT1 related epilepsy, and for all the families in the audience who live this. So I think you have a pretty good understanding. Um, but I think what's important in moving forward is recognizing what gaps there are that do exist. And then I just wanted to talk a little bit, not in high detail, because I think the folks from a variety of different pharmaceutical groups are going to be talking about their individual strategies more specifically, but just to give you an idea of what the sort of strategies and approaches look like right now. So I think when we're thinking about KCNT1 related epilepsy and what your approach to treat the seizures actually is, you have to understand a little bit about what's really going on with KCNT1, what it is, what it does in the brain, and then you can sort of move forward with an understanding of what approach you might need to be able to take to try to address some of those clinical symptoms. So KCNT1 is a gene. It encodes for a potassium channel, a sodium activated potassium channel. And that's sort of schematized here up in the upper right. And this particular protein that is made when the cell tries to utilize the KCNT1 gene assembles itself into what's called a tetramer, right? So it's a channel that has four individual subunits that's sort of schematized here down in the bottom in this 3D model. I don't know if you can actually see my pointer, so I'm going to sort of not bother to do that. So what's actually happening here is this is a channel that sticks inside the outer cell membrane of the neuron. And the way a neuron is going to conduct electrical signals is by utilizing a variety of different channels and causing them to close and open and close. And these channels have to do this in a very precise way, in a very precise combination and orientation and all of that. And it has to do it at the right time and to the right amount. And in the center of this tetramer, when this channel opens, it actually opens a hole that ions can pass through that then change the electrical properties of that cell, cause it to fire or not fire, right? So what happens with these variations in KCNT1 that lead to epilepsy, that lead to neurodevelopmental disorders, is they cause a gain of function problem with the channel. What does that mean? Well, that means that the channel acquires a property that it shouldn't have had in the first place. And the end result is that channel passes too much current. Okay? If the channel passes too much current, then you might say, well, can I slow it down? Can I block that current? Can I then reduce that current closer to something that the cell wants in a neurotypical way to then function properly? And then the downstream effect of that would be, can that correct the seizures? Can that then normalize or improve neurodevelopmental progress, right? So that's sort of, from a biological standpoint, what is going wrong with KCNT1. And that sort of forms the rationale for how might we be able to approach this from a therapeutic standpoint. So on the right-hand side of the slide is a list of all of the current anti-seizure therapies, both medication, dietary therapy with a ketogenic diet, different surgical approaches, different device implants, and actually stole this form from our electronic medical record. I think Jill's sort of smirking and knows. But the idea here is that I like this particular picture because it breaks down all of these therapies based on how they work, right? We have a lot of different drugs that work on sodium channels, right? Things that you're familiar with, right? Oxcarbazepine, lacosamide, phenytoin, etc. Okay. We have a lot of therapies that work on GABA receptors. We have a lot of therapies that have a variety of different mechanisms of action. We sort of classify them as mixed because they work on a variety of different channels. I know it's small, but there's nothing up there that says potassium. That's the K in KCNT1. Nothing. Nothing that we have on the market right now for prescription to be able to provide to individuals who have KCNT1 actually works on potassium channels. That's the problem. Okay. And we published a number of years ago this particular study on the right that sort of looked at in individuals who have KCNT1, and this was the product of sort of the original KCNT1 registry, right? One of those products that came from that initial registry, and the contributions of folks in the room at this point. What we really learned from this is, not surprisingly, there wasn't a lot that was highly effective for the seizures associated with KCNT1. And we didn't even start looking at neurodevelopmental outcomes here, right? This was sort of the first stab at, can we use some of this existing registry data to just get an idea of, are there certain meds that exist currently that might be more helpful for treating seizures? And I think the really end result of this particular study was, I think, something that all of the families in the room really understand on a personal level. There ain't a lot that works well. Right. And when I say sort of here on the left that I've gravitated towards certain therapies in combination, a lot of those things were based upon small signals from this registry data that suggested, hey, keto, it's not great, but it was consistently better than a bunch of other options. Okay. So, to be quite frank, even utilizing different therapies that exist in combinations that seemed to be a little bit better based on registry data doesn't leave you with any smashing successes, right? Seizures are still very, very frequent. Some folks would get better success with their seizure frequency on some of these therapies, but at least from what I've observed clinically, they're really neurodevelopmental improvements with these therapies is minimal to absent, right? Your seizures might get better, but communication doesn't get better. Motor skills don't get better. A lot of the other comorbidities, the GI issues, the sleep issues, they don't get better, right? So there's something fundamentally different about having a KCNT1 variation in your nervous system that is separate from what's going on with seizures, right? And I think at the time we published this study, quinidine was sort of all the rage, right? What was quinidine? Well, quinidine is an old medication that had other uses in the past that folks from drug repurposing studies recognized also kind of blocked potassium channels a little bit. So maybe this would be a therapy that we could then utilize for individuals with KCNT1. And admittedly, there were some folks where it seemed to help, right? And that's what that sort of graph is on the bottom from that paper, right? Some folks had a bit of a seizure improvement. Some folks were temporarily seizure-free, at least in the period of time that we were looking at with this study. But the bars on the left, those are all the folks where it didn't work, right? And I think the reality is at least 75% or more individuals who tried quinidine didn't see a good response. And it wasn't super safe at the doses you had to use it at, right? It caused a lot of cardiac conduction abnormalities, arrhythmias. There were folks who ended up in ICUs related to trying this particular therapy. So it wasn't great and it wasn't super safe. So I think while you'll still see that out there, while there are still folks that will consider trialing it, I think we've in our group sort of moved away from that. in recent years. So I think from a therapeutic approach, there's a few different ways of looking at potential therapies for KCNT1, right? And this idea is this drug repurposing concept that Sarah alluded to at the beginning and that I mentioned with quinidine, right? This is the idea of, can we leverage therapies that are already FDA approved for a different condition that might be useful for individuals with KCNT1, okay? So this particular study that I have up here on the left is a published study on carvedilol, which I think many of you guys in the audience may have heard about before, right? Carvedilol is a medication that was originally intended for folks with heart failure. What does it do? Well, like quinidine, it blocks potassium channels to some degree. It's a heck of a lot safer than quinidine now, right? So the idea is, can we take this therapy that already exists and demonstrate utility in folks with KCNT1? And there's still an ongoing study, I believe, right? Correct me if I'm wrong, Sarah, that hasn't closed yet.
So one patient here, one family who's
So if this is something that anyone in the audience hasn't heard about before and wants to talk more about, I'd be happy to. And you can also talk with the foundation about the observational study that's really looking at the idea of, can we demonstrate, or can a difference be demonstrated with carvedilol? Looking, I think, purely at, or mostly at seizures, but also with some idea of other non-seizure outcomes as well.
there's primarily genetics that we went.
I think the observational study is really focusing on EIMFS, or more severe epilepsy. Yeah. Now, the idea of, would it be helpful for an individual with the sheep phenotype? Well, I don't have a logical reason why not, right? And I think that's something that, you know, could certainly consider from a therapeutic standpoint. But I also want to caution everybody, as Sarah sort of said, I think alluded to at the beginning, the idea of drug repurposing is nice, it's convenient, it's much easier than, say, awaiting the next generation of therapeutics. But I want you to remember that if already existing drug was going to be the silver bullet, we probably know about it already. Okay, so when we're thinking about drug repurposing studies and whether or not they may be helpful for individuals with KCNT1, I think you have to make sure that your expectations are focused on the right things, right? Maybe this will help a little bit with seizures. Will this lead to the non-seizure outcomes that you're looking for from a communication standpoint? Maybe not.
They're not lost.
Right. So, yeah, another approach for KCNT1 novel therapies would be design new small molecules, new drugs that you can take either orally or through a G-tube that will be more precise, right? Make a potassium channel blocker that works well for KCNT1, right? And this is sort of the approach that UCB has taken. This is the approach that Accio has taken, right? With designing novel therapeutic compounds that are specific or more specific for KCNT1 that can block that current and then hopefully lead to improvement in symptoms, right? Now, from a trial design standpoint, these clinical trials are still focusing on primary outcomes that involve seizures, right? Because they're easy to count, they're easy to see. You can do an EEG and find them. And you might have a shot at a highly effective therapy reducing seizures by enough of a frequency to see that signal in the data in the time period the clinical trial is. But small molecules and designer medications aren't the only approach, right? There are genetic therapy approaches that are being investigated, and these are the ASOs or the siRNAs. So what are those, right? So what an ASO is a genetic therapy that really designs, well, let let's take one step back, right? Your genetic code is in your DNA, right? But that message, in order for it to get to a cell in a usable way, has to move through to a protein level, right? Because the proteins are what the cells are actually doing. KCNT1, the channel, that's a protein, right? The genetic message has to get transcribed and then translated and made into a protein in order for that protein to do work in the cell. So in going from DNA to that protein, what the cell is doing is creating what's called a messenger RNA, or an RNA molecule that really represents that DNA code that the machinery of the cell can then turn into a protein. Okay? An ASO therapy, like here in the purple and orange, is a therapy that is designed to stick specifically to that messenger RNA and interfere with its ability to be then translated down into protein. Now, why does this matter? Why might this help? Well, we talked about KCNT1 being a gain of function problem, right? You have too much channel current. How can you interfere with that? Well, maybe you make a medication that blocks the protein channel itself. But maybe you interfere at an earlier stage genetically to try to reduce the amount of messenger RNA that is made, and therefore reduce the amount of protein that is made and turned into a channel. And even if that channel's a little overactive, if there's less of it in the first place, then maybe the sum total of the electrical activity in those nervous system cells is more normal. And therefore you are less prone to seizures and potentially more able to meet your neurodevelopmental milestones at a more appropriate rate. This is the sort of strategy that Servier is taking with their ASO therapy. And then there's this concept, it functions in a little bit different, right? Of siRNA, or small interference RNAs. So these are RNA molecules that get designed to also stick to messenger RNAs in the cell in a slightly different way, that then lead to that individual message either being downregulated or degraded so that it doesn't end up turning into protein. So it's a slightly different genetic strategy with the same end result, right? You're making less protein, so you have fewer overactive channels and therefore hopefully more normal electrical signaling, right? Now, all of these therapies have good preclinical data and very good preclinical data. I think the question that still emerges, and this is the point of clinical trials of these various compounds, is, are they successful in people? Big difference being successful in a mouse or a rat or even a monkey. We're different, right? I think the hope is that any one of these strategies is better than what we have, right? Because what we have, frankly, sucks, if I can use that term in public. The idea here is to just try to come up with something with better promise. And the strategies in the pipeline now look good. I think it's just a matter of what works and what doesn't. And what does works mean, right? Are these benefits for seizures? Are these benefits for neurodevelopment? Is it both? And then the question that sort of plagues me many times is, what are the windows where these benefits might be more helpful? We think earlier is better, but in theory, anybody with KCNT1 should be able to derive benefit from these sorts of therapies if they're able to get them in childhood. So that's what I had for this part of the talk. Questions? Yeah.
I know speaking with Morgan in the past today, so it's like a lumbar puncture, or all the other ones you were talking about the same, aside from they're doing the because they're all going through the central nervous system for a lumbar puncture.
So typically, you're either talking, I believe both of these products are lumbar puncture administered, right? And I think the point that comes up with that is ASO therapies, siRNA therapies, they're not gene editing like you've probably heard about on the news, right? These are therapies where you deliver them, they get into the central nervous system, they have a time window in which they work, then you need another dose. Just like the medication you're taking by mouth or via G tube, right? Has a window where it's effective, then you need it again. So you might imagine sort of the ultimate genetically based therapy is the thing that you get once. Doesn't exist yet.
Is anything working?
In a variety of different conditions, yes, I think folks are moving in that direction because it's pretty obvious that that's more ideal than a lumbar puncture every 3 to 6 months, right?
Sure.
It's a good question. So those sort of therapies, at least the way we understand them now, would have to be delivered to the central nervous system, right? Not IV.
So we have to keep.
Which would be, and the problem with the IV-based therapies is the blood-brain barrier, right? So I know folks have probably heard that term before, but the blood-brain barrier is built into the blood vessel system in the brain and the central nervous system to keep foreign things out. It makes evolutionary sense, right? I mean, at one point in human history, if something got into your central nervous system, that wasn't good. So evolution designed a way to prevent that. But it's also really good at preventing medications that you give through an IV from getting in the brain too. So that problem still has to be worked out.
Theo's not here today, right? We'll be on next after a break. Questions?
Other questions from folks? Yeah.
Why aren't there past starters that
It's
are
they were everyone.
So I think it's probably in part related to sort of an artifact drug design in the way it was done previously, right? Where drug screening protocols were done previously. But remember, all of these ion channels, like sodium ions, potassium channels, these are all also very important in other places, like the heart, right? And safety profiles are going to be different for different ion channels on different organs, right? So it is still a little bit of a mystery for me why we have exactly 0 for potassium. But that's sort of the problem that we're in right now. Yes, absolutely. So the reality is, when I say potassium channels, you think I mean like one thing, right? And the truth is there are many different potassium channels in the brain that are encoded by a variety of different genes, right? With KCNT1 really only being one of them. Now, how some of those other potassium channels lead to epilepsy may be different than how the KCNT1 variants lead to epilepsy, right? This sort of gain of function problem. Some of them actually reduce potassium current and cause epilepsy from that sort of mechanism. But there are others. And blocking those aren't necessarily the solution, right? There was a medication at one point that enhanced potassium current that was called ezogabine, or Potigo was its brand name back in the day, but that doesn't exist anymore for side effect reasons. That actually enhanced potassium current, or would be more potentially problematic if you had KCNT1 because you already have too much potassium current to begin with.